Bacterial morphology. Shapes and arrangements of bacteria. Bacteria occur in various shapes and sizes. We popularly classify bacteria depending on their shape. Cocci. These bacteria are spherical in shape and are seen to occur in different arrangements. Cocci in pairs such as Streptococcus pneumoniae. Cocci in chains such as Streptococcus mutans. Cocci in tetrads such as Gaffkia tetragena. Cocci in clusters such as Staphylococcus aureus. Bacilli. These are straight, rod-shaped bacteria like Bacillus anthracis. Vibrios are curved or comma-shaped rods. Example, Vibrio cholerae. Spirilla take rigid, spiral or helical forms such as Spirillum undula. Spirochetes are flexuous spiral forms. Example, Leptospira interrogans. Other types of bacteria may be cell wall deficient bacteria like Mycoplasma pneumoniae, filamentous bacteria such as Nocardia asteroides. The bacterial cell. A bacterial cell may comprise of extracellular structures such as capsule, Fimbriae, and flagella. All bacterial cells have an outermost cell wall and cell membrane. These enclose the cytoplasm containing ribosomes and other intracellular structures including inclusion bodies, plasmid, and the chromosome. Bacterial capsule. This is a structure that may surround the outside of the cell envelope. It could be diffused, loosely attached to the cell wall in the form of a slime layer. If it is firmly and covalently linked to the underlying cell wall, it is referred to as a capsule. The chemical nature of bacterial capsules is diverse, but majority of them are polysaccharides. These polymers are composed of repeating oligosaccharide units. The capsule is also called as glycocalyx, meaning sugar coat, or a network of polysaccharides. However, the capsule of Bacillus anthracis is composed of a polypeptide, that is, polyglutamic acid. Yersinia pestis produces a capsule of mixed amino acids. Capsules may be weakly antigenic to strongly antigenic, depending on their chemical complexity. They have no net charge and will not bind charged dye particles. Hence, they cannot be stained. Some capsules are very large and absorb water, forming mucoid colonies. Example, Klebsiella pneumoniae. Bacterial cell wall. The layers of cell envelope lying between the cytoplasmic membrane and the capsule are referred to collectively as cell wall. Bacteria can be classified into two types based on the retention of primary dye in gram stain. Gram-positive. When observed under the microscope, gram-positive bacteria retain the primary dye in gram stain and appear blue. Gram-negative. Gram-negative bacteria do not retain the primary dye in gram stain and hence appear pink under the microscope. Gram-positive bacteria. Most gram-positive bacteria have a complex cell wall consisting of peptidoglycan, also called murine mucopeptide. This complex polymer consists of three parts: a backbone consisting of alternating units of NAG, that is, N-acetylglucosamine, and NAM, that is, N-acetylmuramic acid. Tetrapeptide side chain attached to NAM and peptide cross bridges, which are short chains of amino acids that cross-link the backbone. In gram-positive bacteria, there may be as many as 40 sheets of peptidoglycan, comprising up to 50% of cell wall material. Electron micrographs show the peptidoglycan of gram-positive cells to be 20 to 80 nanometers thick. The cell wall also has teichoic acid, which can be of two types: wall teichoic acid linked to peptidoglycan and lipoteichoic acid linked to membrane. Some gram-positive bacteria may lack wall teichoic acid, but all contain lipoteichoic acid. Gram-negative bacteria. The cell wall in gram-negative bacteria includes peptidoglycan, lipoprotein, outer membrane, and lipopolysaccharide layers. Cell wall does not take up any stain. Gram-negative cells consist of a relatively thin layer of peptidoglycan, approximately 10 nanometers. There appears to be only 1 or 2 sheets of peptidoglycan, comprising 5 to 10% of cell wall material. Gram-negative bacteria have an additional outer membrane. The outer membrane is the major permeability barrier in gram-negative bacteria. The space between the inner and outer membranes is known as the periplasmic space, which contains digestive enzymes and other transport proteins. Gram-negative cell walls contain 3 components that lie outside the peptidoglycan layer: lipoprotein, outer membrane, and lipopolysaccharide. Lipoprotein stabilizes the outer membrane by anchoring it to peptidoglycan. Outer membrane is phospholipid bilayer, in which the outer phospholipids are replaced by lipopolysaccharides. It is structurally similar to cytoplasmic membrane and serves to prevent leakage of periplasmic proteins and protects the cell from bile salts and proteolytic enzymes. The outer membranes contain several important porins, which specifically allow transport of solutes. Lipopolysaccharide consists of a polysaccharide core, a complex lipid called lipid A, and a terminal series of repeat units. Each species contains unique terminal repeat units. The polysaccharide core is similar in all gram-negative bacteria. Lipopolysaccharide is toxic in nature and is called endotoxin because it is firmly bound to the cell wall and released only when the cell is lysed. Endotoxin can trigger fever and septic shock in gram-negative infections. Endotoxin can be detected in IV fluids by limulus lysate reaction. Cell membrane. The cell membrane or cytoplasmic membrane is a typical unit membrane approximately 5 to 10 nanometers in thickness. It lies below the peptidoglycan layer of the cell wall and encloses the cytoplasm. It is composed of 60% proteins and 40% phospholipids. The arrangement of proteins and lipids to form a membrane is called the fluid mosaic model. The membranes of bacteria, except Mycoplasma, do not contain sterols. It is a phospholipid bilayer with polar heads on either side of the membrane. Hydrophobic tails are oriented to the interior of the membrane. Specialized structures called mesosomes or chondroids are formed from the convoluted invaginations of cytoplasmic membrane. Mesosomes are more prominent in gram-positive bacteria. They are believed to be analogous to eukaryotic mitochondria since they are rich in respiratory enzymes. The bacterial chromosome is attached to the septal mesosome. During cell division, the septal mesosome participates in the formation of cross walls. Cytoplasm. The cytoplasm or protoplasm is the portion of the cell that lies within the cytoplasmic membrane. It is gel-like in consistency and includes the prokaryotic chromosome and ribosomes. Constituents of cytoplasm include proteins, including enzymes, vitamins, ions, nucleic acids and their precursors, amino acids and their precursors, carbohydrates and their derivatives, fatty acids and their derivatives. The cytoplasm does not exhibit any internal mobility, that is cytoplasmic streaming. The cytoplasm also lacks organelles such as mitochondria, Golgi apparatus or endoplasmic reticulum. Genetic material. The chromosome in bacteria is typically a single, closed circle DNA that is concentrated in a nucleoid region. It is not membrane-bound as in eukaryotes. Some bacteria also possess smaller extrachromosomal pieces of DNA called plasmids. Plasmids replicate independently of the chromosome and carry genes that are not essential for cell survival, but may give some advantage to an organism. The chromosome is attached to an invagination of the cytoplasmic membrane called mesosome. Mitotic apparatus and nuclear membrane are completely lacking. The length of E. coli chromosome is approximately 1.4 millimeters but is condensed inside the cell by supercoiling. DNA is mainly negatively charged, hence bind readily to basic dyes. It can be demonstrated by Flugen stain or by electron microscopy. Ribosomes. The distinct granular appearance of prokaryotic cytoplasm is due to the presence and distribution of ribosomes. Bacterial cells can contain thousands of ribosomes, which are the sites of protein synthesis. Often, they aggregate to form structures known as polysomes. Bacterial ribosomes are termed 70S, that is Swedberg units, and eukaryotic ribosomes are termed 80S. The difference between bacterial and eukaryotic ribosomes is often exploited during antibiotic therapy. Inclusion bodies. Intracytoplasmic inclusions can be vacuoles, crystals, or storage bodies. Bacteria often store reserve material in the form of insoluble cytoplasmic granules. Inclusions accumulate when a cell is grown in the presence of excess nutrients, and they are often observed under laboratory conditions. Flagella. Some bacteria are motile and some are not. Almost all motile bacteria possess flagella as the organ of locomotion. Motile bacteria tend to move towards or away from a source of stimulus. The stimuli can be chemicals, that is chemotaxis, light, phototaxis, air, aerotaxis, or magnetism, magnetotaxis. The flagellum comprises of three parts: filament, hook, and basal body. The flagellum is attached to the cell body by hook and basal body, while the hook and basal body are embedded in the cell envelope. The filament is free. If a flagellum is cut off, it will regenerate until it reaches a maximum length. The different types of flagellar arrangements are: monotrichous, a single flagellum at one pole, also called polar flagellum. Example, Vibrio cholerae. Amphitrichous, single flagellum at both poles. Example, Spirilla. Lophotrichous, two or more flagella at one or both poles of the cell. Example, Spirillum undula. Peritrichous, completely surrounded by flagella. Example, E. coli. Fimbriae. Fimbriae or pili are short hair-like structures made up of protein pillin and are present in many gram-negative bacteria. Fimbriae are shorter and straighter than flagella and are more in number. They are 0.5 micrometers long and 10 nanometers thick. Even though pili arise from the plasma membrane, they are not considered part of the plasma membrane. They are anchored in the membrane and protrude through the cell wall to the outside of the cell. Since they are made up of protein, they are antigenic. Bacteria from different genera may possess common fimbrial antigens. Fimbriae are usually seen in young cultures and lost on subcultures on solid media. Bacterial endospores. In poor growth conditions, some bacteria such as Bacillus produce resistant survival forms termed endospores. This process is known as sporulation. Bacterial spores are endospores in contrast to fungal spores, which are usually exospores with reproductive function. The DNA is replicated. The cytoplasmic membrane invaginates, forming a spore septum within the cell. Then, forespore is formed. The forespore is engulfed. The forespore forms a peptidoglycan cortex, and a spore coat is added. The mature spore is released from the mother cell. The presence of the cortex and coat makes endospores resistant to extreme environmental conditions like high temperatures, dryness, UV radiation, and toxic chemicals such as disinfectants or antibiotics. The endospore is able to survive for long periods of time until environmental conditions again become favorable for growth.